Welcome back to our channel. Today, our second class of microbiology. Today, we will discuss about the bacteria, and so that it is come under bacteriology. We are discussing about the bacteria, so that it's called bacteriology, and we are just starting the microbiology as well as the core microbiology from today's class. Just stay tuned with me. You will not face any kinds of doubt after completing this class. Bacteria is very small, so that it is microorganism, right? Microorganism. We cannot see into our naked eye, so that we are saying that it is a microorganism. See, if it is bacteria, if the bacteria is very small, as well as if the bacteria is round in shape, that is called cocci. If there is two bacteria round in shape is present, that is called diplococci. If there are two bacteria are present in round shape, that is called diplococci. Okay. If the round shape bacteria are arranged in a chain, that is called strepto. That is called strepto. They are chain like things. That is strepto. Streptococci. Why it's cocci? Because it is round in shape. And why is strepto? Because they all are the chain in nature, right? If they are arranged in a grapes-like structure, that is called staphylococci. Why it's cocci? Because it's round in shape. Why it's staphylo? Because they all are arranged in a grapes-like structure. After that, if I am coming the things, that's all are regarding cocci. Now I am coming to the rod section as well as rod shape bacteria. If the bacteria is rod in shape, that is called bacilli. Understood? That arrangement is called bacilli, but when the bacteria we are saying about the bacteria should be bacillus. After that, if the rod shape are not proper, if the rod shape are little bit round shape, that is called coccobacilli. Coccobacilli. If there is the bacilli are arranged in a chain, that is called streptobacilli. Streptobacilli. Understood? That's all are regarding the bacilli. After that, there is a bacteria that is coma shape. That is called Vibrio, which basically causes the cholera disease. Cholera is caused by Vibrio cholerae. That is also bacteria that is coma in shape. There is also one bacteria that is Chinese letter shape, that is called C. diphtheria. That is Corynebacterium diphtheria, which basically causes the diphtheria disease. So that's all are the arrangement of the bacteria. Due to the arrangement or the arrangement wise, the bacteria are dividing in this way. That's all are the arrangement. Hopefully you have understood the arrangement, so that if I am sum upping these things, there is a cocci, there is a bacilli, there is a vibrio, there is a corynebacterium diphtheria, or we can say that corynebacterium, that is Chinese letter in shape. That's all are the arrangement of bacteria. If I am coming to the structure of bacteria, I am just erasing the arrangement. So that you can easily understand the things properly. It will help you to understand the whole things. See, if I am coming to the bacteria, see here, there is a normal bacterial shape. It is a normal bacterial shape, right? See the structure of bacteria. If you are able to understand the structure of bacteria, you can easily understand the whole bacteriology basic things. Okay, and whenever you will understand about the basic things about the bacteria, you can easily understand the systemic bacteriology, where you will understand the every bacteria in one chapter. That is systemic bacteriology. You will understand it later. Firstly, you have to complete the general bacteriology or general microbiology. After that, you have to move there. But here is the basic structure of bacteria that is important for you guys, that is important for the biological students as well. That is the basic structure. See, completely outermost layer, that is called capsule. That is capsule, right? Completely outermost layer, that is capsule. Now I will say you that one kind of substance I will take from here, and I will describe into this board about this substance. See here, capsule basically not mandatory for every bacteria, not M for mandatory, not mandatory. Some kinds of bacteria basically prost, and some kinds of bacteria basically contains with this capsule. And now what is this capsule? Capsule basically one viscid secretion viscous secretion from the cell wall. This secretion when takes the structure, that is called capsule, and whenever this secretion will not take the structure, that is called slime layer. Understood? That is called slime layer. Okay, so that we have understood the capsule, and the function is capsule basically here to protect the bacteria. Capsule basically have the virulence, so that if any kinds of antibacterial agents are coming, capsule help to fight it first, and capsule have their virulency. Understood? That is capsule. Secondly, if I am coming to the second structure, that is the cell wall. See here, cell wall. Here is the cell wall. We will discuss about the cell wall right now. See, cell wall basically help to protect the bacteria. P for protect the bacteria. See, and second one, without the cell wall, your bacteria will not taking the structure, so that it basically help to giving the shape of the bacteria. This cell wall also help to the division of the bacterial cell division. This cell wall are help to protect as well as to giving the eternal protection of the bacteria. This cell wall. After that, this cell wall basically preserve the internal things as well as creating the barrier from outermost side and the innermost side, so that the cell wall should be semi permeable. Understood? So that's all about the cell wall. Now my question is, how to the cell wall is formed? There is a two types of cell wall, one for gram positive and one for gram negative. I will take the another class for the gram positive and gram negative bacteria. You will understand how the cell wall is look like, as well as how is the arrangement of the cell wall. You can easily understand. I will take the class for gram positive and gram negative bacteria, because within this short class it is not possible to discussing the cell wall briefly. But I am saying you that this cell wall are composed with lipid, this cell wall are composed with the protein. Okay, and this cell wall here might be present the teichoic acid, here might be present the polypeptidoglycan. Okay, and we will understand in the next class in gram positive and gram negative bacteria. Okay, that is the cell wall. After that, see, here is a ribosome basically present. See here, inside the cell, that is ribosome basically without cell wall, without cell wall, without wall. Ribosome have no membrane. See here, is there any kinds of membrane? Is there any kinds of membrane? No, there is no membrane. So the ribosome cannot contains any kinds of membrane. Please keep in mind that. And bacteriology or bacteria is the prokaryotic cell, not eukaryotic. Prokaryotic cell, because that is the cell and that is the structure of bacteria that basically contain with one cell, so that it is called the prokaryotic. Bacteriology as well as bacteria is not eukaryotic, it is a prokaryotic cell. Please keep in mind that. Here is a ribosome. I have discussed with you that ribosome should not contains any kinds of membrane, and ribosome basically help to protein synthesis. We have understood into the structure of the cell. If you are relating the bacterial cell with the normal cell, then you can understand that. What are the difference between the prokaryotic cell and eukaryotic cell? Prokaryotic cell defining the bacterial cell and eukaryotic cell defining our human cell. There have lots of cell, more than 200 cell. That is human cell, the RBC, WBC, platelet, fat cell, hepatocyte cell, Kupffer cell, lots of cell, right? They all are the eukaryotic cell. Understood? So the ribosome we have understood. After that, I am coming to the mesosome. There should be also some kinds of minerals. See how the mesosome is formation is occurring. Mesosome, how the mesosome formation is occurring. See, the cell wall, and within the cell wall there should be cytoplasmic membrane. The cell wall become consist and become contract to form one type of vesicle like structure. See here, it is one type of vesicle like structure, right? It is called mesosome. Now, sir, what is the function of mesosome? You need to understand the function. Function basically nothing but this mesosome help to the respiration. We are human beings, we also require the respiration. Bacteria is also require the respiration. They have some kinds of aerobic bacteria, they require oxygen. They have some kinds of anaerobic bacteria, they require carbon dioxide. We will understand this kind of things later. Okay, and there have some kinds of bacteria, facultative aerobic, obligate anaerobic. There have lots of another terminology. Into the next class I will understand and I will explain these things. But what will you do? You have to subscribe this channel, otherwise it is not possible to giving the classes in every day, because it takes lots of effort. Without your support or without your subscribe or without your tendency of education or without your hungerness of learning, I can't give you the things. See here, we have understood the mesosome, right? After that, if I am coming to the nucleus, see here, that is the nucleus. What is nucleus? Nucleus basically called nucleic acid, that basically called carrying the DNA or RNA. Almost bacteria contains with DNA, that is deoxyribonucleic acid. And one thing, if you are noticing here, into the human beings or into the eukaryotic cell, if you are noticing, nucleus have the membrane, right? There was the membrane, there was the membrane in eukaryotic cell, but into the bacteria, see here, that is naked nucleus, that does not require any kinds of membrane. If you are noticing here, see here, that is the DNA, there is not present any kind of membrane. That is the difference between the eukaryotic cell and the prokaryotic cell. These questions are too much askable question into your competitive exam as well. See, nucleus have no membrane in for bacteria, or for bacterial morphology, there is a no membrane in nucleus, so the nucleus is naked. This DNA or nucleus help to carrying the genetic materials, as well as this basically help to create the bacterial genome, so that some kind of characteristics, some kinds of genetical features, they basically help to carry to the future generation. Understood? That is the nucleus. After that, we have understood. So that which kind of things we have understood? See here, we have understood the capsule, we have understood the ribosome, we have understood the cell wall, we have understood the DNA. We have to understood the flagella. It is also an important thing. See here, if flagella, what is the function of flagella? Firstly, you have to understood that what is flagella. Flagella is one type of thread-like protein. Flagella is composed with flagellin protein. See here, if you are noticing here, flagellin. Flagella is composed with flagellin protein. Understood? And this flagella basically help to move. Help to move. So that motility, if their flagella is present, this bacteria should be motile, and if the bacteria does not contain any kinds of flagella, that is called non-motile bacteria. Understood? Without the flagella, the bacteria cannot be moved. Flagella basically help to movement. Understood? And there have also lots of arrangement of flagella. I will take the another classes for this one, because bacteriology as well as microbiology is too much vast. Okay, too much vast. We need to understand a little bit. Understood? That is the flagella, and without flagella the bacteria cannot be moved. And last but not the least, that is the pili. Pili is also one type of thread like structure, and pili is composed with pilin protein. See here, pilin protein. Pili is composed with pilin protein. This pilin protein or pili basically help to conjugation. Conjugation means whenever these bacteria are attached with another bacteria, they basically help to transport a genetic materials. See here, transport the genetic materials, so that here required the pili. Without the pili, the conjugation cannot be happening. So that pili's function is to conjugation. Pili's function is to attachment. Pili's function is to transfer the genetic materials. Understood? That's all at pili. Hopefully you have understood the whole morphology. I am again saying you that that is a basic class. You cannot able to understand everything, as well as you can take a little bit idea regarding the bacteriology. If you want to understand a huge amount of this kind of things, just check our description box. There have the classes links, as well as there have the batch link. Firstly, you have to join our free batches. After that, if you are attracted with our study as well as with our pattern of study, then you can move. Okay, and up to this much, I hope anyone not facing any kinds of doubt, so that if I am sum upping these things, we have understood that the arrangement of bacteria, Cocci, Bacilli, Vibrio, and as well as the Corynebacterium. After that, we have understood the general bacteriology as well as the general structure of bacteria, that is capsule, cell wall. After that, if I am coming to the innermost portion, there should be the cytoplasmic membrane. After that, ribosome. After that, DNA. After that, mesosome. After that, flagella. After that, pili. So that hopefully you have understood everything. It's my humble request to all of you, please share this video in the maximum amount, so that it will help me to inspire, it will help me to creating new video for all you guys, and it will help me to giving the free classes for everyone. Okay, so that please spread this video and check the description box, only for you guys, only for the dedicated and the passionate medical laboratory technologists. We have designed a M.L.T free batch, so that you can easily join there and understand lots of new things regarding M.L.T. Thanks a lot for joining.